Test for proteins. We go for the Biuret test. We're supposed to take a small quantity of fresh milk solution. We have it already in the beaker. We take small quantity of it. We take another we have to take a small quantity of fresh milk solution. Then we add 1 ml of sodium hydroxide. This is sodium hydroxide. We add it up. You have the sodium hydroxide, and then we will now have a drop of copper 2 tetraoxosulfate 6 solution. This is copper 2 solution. We'll still add to it. Now let's see. You have to keep observing. We shake, and what color are we seeing? The mixture is changing to purple. This is purple. So when the mixture turns into purple, it shows that protein is present in the milk. Can you all see the purple color? So this signifies that proteins is present in fresh milk. We use fresh milk solution. We added 1 ml of sodium hydroxide, and we added a drop of copper 2 tetraoxosulfate 6 solution. And we have seen the change in the color. It was initially milk. It turned into purple after the addition of these reagents. And this infers that protein is present in fresh milk. Is that okay? Good. Now we test for oil. This is a filter paper. You can use any available white paper. We have oil here. We drop some of it on the paper. We just have some few drops of oil on a white paper. This is a filter paper, very common in the lab. And then the translucent test allows us to view this paper through any available source of light. We look at it through the window, and you can see the spotted parts looking transparent. We look at it through any available supply of light. We look at it through the window, and we can see the center where we have the oil spot looking transparent. Now this transparency is what we refer to as the translucence. This is the translucent test, and it shows that oil is present. We have gone through certain food substances in our houses, common ones, which shows that these materials are present. We have run a simple test using starch material, bread, to see the presence of starch. We have tested for simple sugar in glucose. We have used the laboratory glucose. At home, we can equally use the cane sugar juice. Cane sugar. You can collect the juice and you can run this simple test and you can have this particular result. So if you don't have the laboratory glucose, you can use simple cane sugar, which is very abundant in our society. We have seen the test for protein where we use fresh milk solution plus sodium hydroxide and copper 2 tetraoxosulfate 4. We have tested and we have seen the presence of these food materials in this food process. So let's go back to the table. The first column, test for starch. Collect starch material. It could be bread. But for this particular class, we have used the bread. It could be yam. It could be any other starchy food. But for this purpose, we have used bread. We diluted it. We put some water in it to have a solution. We added few drops of dilute iodine solution. And we have seen the reaction. The color changed into blue-black. That is what we observe. And that observation refers that starch is present in our bread. We have put a small quantity of glucose solution. We used the laboratory glucose. We diluted it, we dissolved it to have a solution, and then we added the Benedict solution, and we boiled, we heated it using our source of heat, the Bunsen burner, and then we had an orange precipitate being observed. The orange and the brick red gives the presence of glucose. We have tested for proteins. We took a small quantity of fresh milk. We added sodium hydroxide solution and copper 2 tetraoxo 6 solution. We have observed the mixture turning into purple. And then that purple color tells us that protein is present in the milk. We have equally done the paper test, translucent tests. We tested for fats and oils. We have some drops of oil. This is vegetable oil. We had the drops and we looked through the available source of light, and the transparent part shows that oil is present. So it is very important that we know these things, we carry them carefully and gradually, and we have our best result. If we keep to the instruction given, and the examiner will definitely give you the table. Test for a particular thing gives you the procedure, and now it is left for you to follow the procedure strictly and then observe what will come out of it. Your observation gives you your inference. That's okay for now.